Hello everyone, here we are with part 2 of 11.4 sexual reproduction, and we're going to go through the details of spermatogenesis and oogenesis. There are some major differences between these two gametogenesis events that you need to know about for the IB. So with spermatogenesis, before we were talking about how it occurs in these seminiferous tubules within the testes. This is a cross section of that tubule. Okay, we're starting with the spermatogonia. Those are going through mitosis to divide, and then some of the spermatogonia go on to go through meiosis, meiosis 1 and meiosis 2, in order to make the spermatids, and then finally the spermatozoa. So how do they actually go about doing that? We start with the spermatogonium. These have 2n number of chromosomes, aka 46 chromosomes. They go through mitosis, which is making an exact copy, as you know. That's what mitosis does. So we've got these other cells, which are known as primary spermatocytes at that point. Okay, these still have 46 chromosomes. Then we go through meiosis 1, that halves the number of chromosomes, and now we have these secondary spermatocytes. We then go through meiosis 2, and that's where we finally have the spermatids. Okay, we still have half the number of chromosomes here, but now we have 4 spermatids due to meiosis. Those spermatids are almost full-fledged sperm cells or spermatozoa at that point, but they do need some extra nourishment from those Sertoli cells that we talked about before in the seminiferous tubules, and that's going to help them to continue their differentiation into full-fledged sperm cells, which are then released into the lumen of the seminiferous tubule and can then go to the epididymis, right, that stores the sperm cells, and then through the vas deferens, which is the sperm duct that carries the sperm out. Important things to note about this is that it's going to look different from oogenesis in a few different ways. So first of all, there are 4 cells as the end product. That is not the case in oogenesis. You'll also notice that they're quite small and they have smaller amounts of cytoplasm than in oogenesis. Another major difference is that as these cells are going through mitosis and then meiosis, this is just a continuous process. There's no pausing that happens throughout any of this, which does happen in oogenesis. Here's a more detailed image that shows you some of the more details of what's happening here. So remember, mitosis produces more spermatogonia. These are those parent cells that do need to reproduce in order to make the primary spermatocytes. Some of those spermatogonia do grow to become the primary spermatocytes. Okay, note that you do not actually need to know about the roles of FSH and LH in males, but it's kind of interesting to know that there are these same hormones that are used in females for sexual reproduction and during ovulation. Okay, remember Felt-Popo. These same hormones are actually being used. Okay, but don't worry about remembering that they are also used in spermatogenesis. Just an interesting thing to know about. Okay, spermatocytes do then end up dividing by meiosis, these primary spermatocytes that came from the spermatogonia. And then we've got 2 cells here. But those 2 cells divide again, just like you know with normal meiosis, to produce this 4 haploid spermatids. Okay? Then you've got testosterone, right, being produced. That's going to help to differentiate the sperm. Part of the differentiation that happens here is tail development, the midsection with that mitochondria forms, and then you've also got nourishment from the Sertoli cells. A quick review question of that. Through which process does a spermatid become a fully functioning spermatozoan? And you can pause here before I say the answer. So it would be through differentiation with the help of testosterone. Now we're going to move on to the ovary and how oogenesis creates new egg cells in the ovary, and how does this differ from spermatogenesis? That's the important key here, is you've got to really take note of these differences that are happening. Something interesting that's different between spermatogenesis and oogenesis is with oogenesis, we have these pauses that are happening throughout meiosis. So meiosis will pause at different times and then resume at other times, versus spermatogenesis, where meiosis 1 happens and meiosis 2 happens, and we have a constant production of sperm cells. In females, we've got the oogonia, which are the equivalent to the spermatogonia. Remember, like the parent cells that produce those egg cells. The oogonia are going to be producing the primary follicles from birth. So females all contain their primary oocytes, okay? These are the equivalent to the primary spermatocytes in spermatogenesis, okay? They contain all of these from birth, thanks to the oogonia that go ahead and start replicating and make tons and tons of these primary oocytes. So to get a general idea of what this looks like, here would be an oogonium, just like the spermatogonium. That is 2N, so it has 46 chromosomes. It goes through mitosis, just like the spermatogonium, and it's going to make the primary oocyte, just like the primary spermatocyte. This primary oocyte, there are millions of these in females from birth. However, the thing that's different between males and females is the primary oocyte will just stop right there, and it will not go through meiosis 1 and then meiosis 2 until later on. Okay, so once a female reaches puberty, some of her follicles, right, with the primary oocytes within them, are going to be developing further in response to that hormone FSH. Okay, the oocyte is going to complete its first meiotic division, and then it's going to be producing a secondary oocyte, as well as what's known as a polar body. So this is different than in spermatogenesis, because remember, in spermatogenesis, right, we have the primary spermatocyte, and then it produces 2 secondary spermatocytes. But in oogenesis, we have the production of 1 secondary oocyte and 1 polar body. This oocyte, the secondary oocyte, again halts its meiosis processes. It halts at prophase 2 until the secondary oocytes continue to develop with the follicle further, and once the follicle that's surrounding it is mature enough, it's going to rupture to release the secondary oocyte with a small number of cells, which is the mature egg, into the fallopian tube. Okay, this is then going to finally go on to complete meiosis 2, the final formation of the ovum or the egg. Okay, so it goes through meiosis 2, and then we have your final ovum, final developed ovum, as well as another polar body. So what are these polar bodies all about? Well, these are what would be another egg cell. However, it's actually more advantageous to put all the nutrients and resources in cytoplasm into one large viable egg, and this is where the differences in cytoplasm come about as well, because remember, in spermatogenesis we had smaller amounts of cytoplasm but more of the cells that are produced, more of the gametes. But here we have less gametes being produced but more cytoplasm and nutrients. So this is actually advantageous because we want an egg with a really viable egg with all of the nutrients available and cytoplasm available for that developing embryo and for fertilization to occur. And so then the same thing happens when we go through meiosis too. We produce that final ovum with a large amount of nutrients and cytoplasm and another small polar body. So again, that sort of would be an egg maybe, but it's a small egg that's going to eventually degenerate because it's better to have this more viable, larger ovum with more cytoplasm and nutrients. The end result of all of this is one final viable egg. Versus in spermatogenesis, we have the 4 sperm cells that are produced. As a general recap here, females are born with all of the primary follicles that they would need, including the primary oocyte inside of there, okay, the primary egg cells. And then they continue to develop into the secondary oocytes and the mature follicle that's surrounding it and supporting its development. Then eventually ovulation occurs, okay? The egg cell is released, and so that would still be the secondary oocyte at that point. It goes through another round of meiosis, the last round, and produces the final ovum or egg. Okay, and so you can practice your annotation of the diagrams of the ovary as the IB would like you to have done. You can pause here, and then I will show the answers on the next slide. So here we have the germline epithelium where the primary follicles are going to be generated. And remember, all the primary follicles are present at birth in a female. They're going to, around puberty, be developing into the mature follicles, okay, and meiosis 1 would be happening to produce the secondary oocyte. This was the primary oocyte, this is the secondary oocyte. You can see the zona pellucida is there as well. And then eventually ovulation occurs. The secondary oocyte is ejected into the fallopian tube, and the second round of meiosis 2 would occur at that point, where we would have the fully matured ovum ready for fertilization. Here are some examples of questions, past IB questions that you could use to help you study. So pause here, try to answer them, and I will show the answer on the next slide. Here's the answer for part A. Pause here. Here's the answer for part B. You can pause here. Now again, remember we're trying to focus on that comparison between oogenesis and spermatogenesis. So these are all of the differences that you may have noticed. You'll notice that they are produced, the gametes are produced in different locations. You'll notice that when the process starts or is initiated is going to be different as well. In females, remember, it's during development of the embryo or fetus. Females have all of their primary oocytes from the moment they were born. In spermatogenesis, that's not going to start in males until puberty. Okay, there are breaks during meiosis. Remember, in females, there are no breaks and no pauses in meiosis during spermatogenesis in males. Okay, cytokinesis is not really equal in oogenesis. We're trying to create these larger cells with more cytoplasm for the eggs and more nutrients, versus in spermatogenesis there's equal division and we're creating the 4 sperm cells versus the 1 egg cell. So that's the other difference. The number of gametes that are produced or released, right, is going to be different. Oh, you should also note that there are polar bodies being produced, right, in meiosis with oogenesis. Okay, the timing of release for males, the sperm are constantly being produced and released, versus females, only being released on the 14th day of their menstrual cycle. Okay, in gametogenesis, does it ever stop? It does stop in females at, this is called menopause. Okay, but in males, that's going to be continuing throughout their whole adult life. And also remember, you need 8 of these points to completely get those marks. Here's another example of a similar question where you compare and contrast these processes. You can pause here, and I'll show the answer on the next page. Here you go. Okay, this next part is going to be about fertilization of egg and sperm. One of the things that the IB wants you to know about is that fertilization in animals can be internal or external. So there's internal fertilization and external fertilization. Let's start with external. Some aquatic species, like fish and amphibians, practice external fertilization. What does this mean? It means that the female will lay her eggs, and shortly after, the male will release his sperm into the water, and the eggs are fertilized. This can increase the total number of offspring, okay? But this also is susceptible to a lot of environmental variation, like changes in the water temperature or pH, for example, or maybe even pollution, and also predation. So that's going to lead to an overall greater loss in gametes or offspring. Versus internal fertilization, because it is internal, you actually don't have to produce as many gametes, so you don't have to produce as many egg or sperm, and it's in more of a contained environment. It's going to prevent dehydration of the gametes as well, and that's why this mainly occurs in terrestrial organisms. Now, when we get into the details at the molecular level and the cellular level with egg and sperm and how do they fertilize each other, these are the kinds of questions that you want to be focusing on. Why can only one single sperm fertilize an ova, or an egg? And what prevents polyspermy? Now, I did mention this before when we did the diagrams. Another question you'll want to be thinking about, how does the sperm penetrate the zona pellucida? Again, I mentioned this when we drew the diagrams of the sperm and egg. Okay, last thing you should be thinking about is what causes the pause in the ova's meiosis process to continue. The diagram of fertilization occurring, as you can see, the sperm is going to approach the egg. Okay, the head starts to push into the zona pellucida. Their membranes actually do fuse, the sperm membrane and then the membrane of the egg. But before that, the acrosome, remember there's that acrosome here at the head of the sperm, that contains what again? And is used for what? It contains the enzymes that is used to digest and break apart the zona pellucida so that it can then have its membrane fuse with the egg's membrane and eject its genetic information into the cytoplasm of the egg. Remember that there are these cortical granules that are doing something here as well. They're going to be ejected into the zona pellucida through exocytosis, and they're going to be hardening the zona pellucida. Why again? Why are they hardening the zona pellucida? To prevent polyspermy. We only want one sperm to be entering the egg at a time. If more than one are going to be entering, then there's not going to be a successful fertilization. Okay, and so here you can see the membrane of the sperm and ova fuse. This does stimulate exocytosis of these cortical granules that then have that then cause a hardening of that zona pellucida. Okay, and then you'll notice this answers that last question that we were talking about before. There's an influx of calcium into the ova, which prompts the completion of meiosis II. So what does that tell you? Well, basically, meiosis II has not occurred yet at this point. So fertilization is actually what prompts the egg to go through meiosis II and then to form the complete ova that it needs to form. And of course, finally, we know that once the nucleus of the sperm cell is deposited into the cytoplasm, it's going to fuse with the ova's nucleus to form that diploid zygote that's going to start developing into a baby. Here's a question that you can use to help you practice. Where does the acrosome reaction occur? You can pause and I'll tell you the answer. So it should be occurring at point C, right? So that's where the zona pellucida is. Remember, the acrosome reaction is the enzymes breaking down the zona pellucida.